Hello everyone and welcome to this webinar. I have the privilege of describing to you the many new enhancements to GastroPlus in version 9.8. This is a version that was not going to exist until the science and scientists demanded that it had to be released. I'm sure you're aware that we have a team of specialists dedicated to rolling out GPX, which will be a new user experience in PBPK modeling. But in parallel, another team of scientists were adding many new features to version 9.7, which led to the birth of version 9.8. Quite simply, we wanted to deliver this science to you today. I will go through the many developments, which you can see from this slide here. A lot of them are focused on the additional dosage routes module independently. So let's start with the ADMET Predictor module. The recently released ADMET Predictor version 10 APX contains a new transporters module that sees the increase in transporter models from 6 to 24. In addition, the new models include the prediction of the Michaelis constant Km for 6 uptake transporters: OATP1B1, OATP1B3, OAT1, OAT3, OCT1, and OCT2. Now, there are no Vmax predictions, so the Km values have to be manually added to the transporter table within GastroPlus, but they do provide good initial estimates for PBPK models. As you can imagine, the addition of so many extra predictions required a restructuring of the GastroPlus notes. And at the top of the screen here, you can see a screenshot of the transporters section within the new GastroPlus notes for version 9.8. Another enhancement is based on an internal verification. The default clearance model when importing structures has now changed to the total HLM from the all RCIP model. Now let's focus on the ACAT model enhancements. Firstly, you can now mechanistically model cyclodextrin formulations. What are cyclodextrins? Well, cyclodextrins are cyclic oligosaccharides that are pharmaceutical adjuvants. The molecular structure of these glucose derivatives, which approximates a truncated cone, bucket, or torus, generates a hydrophilic exterior surface and a nonpolar interior cavity. Cyclodextrins can then interact with appropriately sized drug molecules to yield an inclusion complex, which can be used to enhance the aqueous solubility, physical chemical stability, and bioavailability of drugs. However, the free drug concentration can be much lower than in conventional formulations, and this will impact the rate of absorption and precipitation. And I've got an example in a couple of slides that shows that. To access the cyclodextrin effect, you would either use the quick link toolbar within GastroPlus or go via simulation setup number 5 dissolution model. From the screenshot here, you can see that when including the cyclodextrin binding effect, 3 parameters are required: the cyclodextrin dose in mgs, the cyclodextrin molecular weight, and the K association constant in liters per mole. There are a few key points that I would like to make for the cyclodextrin effect. The first one is that it is for any oral immediate release dose. It is not available for other dosage routes. And it will automatically be switched off if you select one of these other dosage routes and the include cyclodextrin binding effect was switched on because previously you were modeling an oral immediate release dosage form. Another point is that drug can be dissolved and not available for absorption as it's bound to the cyclodextrin, and it's only the unbound drug which is permeable and available for absorption. Cyclodextrin itself is not absorbed, and you can really nicely show this with a new plot select curves functionality. So you can show the cyclodextrin transitioning through the 9 ACAT compartments and ultimately being excreted. The final point is that the solubility absorption interplay can slow down intestinal permeation of compounds in the presence of cyclodextrin. And as I said, I will show you an example in a couple of slides' time. The new, or the inclusion of the cyclodextrin formulation means that we've now got a new tab in select curves, a new plot select curves, and that enables you to plot the details of the dynamic binding to the cyclodextrin. You can see here that we've got up to seven new plots, and these cover cyclodextrin and the drug bound and the free amounts and concentrations. Let's move on to that example. So here we have a BCS class 2 weak base. pKa, a basic pKa of 4, a lipophilicity, a log P of 4, aqueous solubility of 1 microgram per mil, a pH 7, and an effective permeability of 1.5 times 10 to the minus 4 centimeters per second. On the left-hand side, you can see that cyclodextrin effect is not on. No cyclodextrin with this simulation here. We have a fraction absorbed of 42%, a Tmax of 0.32 hours, and a Cmax of 0.73 microgram per mil. From the red curve, the percent dissolved, you can see we're 100% dissolved. We're dosing a solution here. There's precipitation up to about 80% almost of the dose. If you look on the right-hand side, this is where we have included the cyclodextrin effect. So this is with cyclodextrin. And the fraction absorbed has increased to 93%. The Tmax has slowed to 0.8 hours, and the Cmax has increased to 1.12 micrograms per mil. So this solubility absorption interplay has enhanced the exposure. It's slowed down the permeation. You can see from the percent absorbed, the sort of cyan-colored curve is a little bit slower in terms of going up the curve than on the left-hand side here. We've got the same scales in both plots. But the cyclodextrin effect, although it slowed down that permeation, has reduced the precipitation. The dotted red line here is a lot lower on the right-hand side than it is on the left-hand side. In terms of verification, here we have a poster from Robert et al. Which shows the initial verification of the inclusion of cyclodextrin formulations within GastroPlus, and we look forward to hearing your verification of this new functionality within GastroPlus 9.8. Still with the ACAT model, but now we're going to talk about the simplified conversion of metabolite back to parent in the gut lumen. So what we're talking about here is acyl-glucuronide conversion to parent in the gut lumen. Now, previously, when modeling the enterohepatic recirculation of a glucuronide, the conversion back to parent had to be in the enterocytes and required a high activity enzyme in the gut. Now, there's a new checkbox on the EHC window which allows for simple conversion of a glucuronide back to the parent in the gut lumen. See the checkbox here. This is not a general solution for compound metabolism in the gut lumen. That's coming in GPX. It's applicable here primarily for acyl-glucuronide metabolites. It produces parent compound in the lumen for reabsorption, and it assumes a breakdown of glucuronides, so the molecular ratio is 1. Moving away from the ACAT model and now onto our PBPK model, I want to talk to you about the extension to the lung tissue in a PBPK model. So working very closely with one of our key partners, we've updated the lung tissue in our PBPK model to include partitioning into the epithelial lining fluid, the ELF. I must emphasize that this extension applies to the general lung tissue in the PBPK model, and that the mechanistic lung model in the pulmonary compartmental absorption and transit, the PCAT module, already included the lung fluid compartment. The driver for this extension was for a fit-for-purpose model for drug discovery, where partitioning into the ELF following oral dosing needed to be predicted, but our PCAT module was too detailed and required too many inputs for compound identification and prioritization, which is the focus of drug discovery. So this extension involves the addition of an ELF compartment to the apical side of the lung tissue, and a compound can partition via an ELF unbound plasma partition coefficient, or we're looking on the left-hand side here at perfusion-limited tissue model, or it can transition as a combination of slow diffusion. So this is through the permeability surface area product, the PSTC, or carrier-mediated transport as parameterized by Vmax and Km. For a permeability-limited tissue, It can be by that slow diffusion or carrier-mediated transport. So it depends on the tissue type and the settings as to how the compound would partition into the ELF. The ELF unbound plasma partition coefficient is a user-defined value. And the initial estimate is available with the license to ADMET Predictor module. Now, this model won't be available in ADMET Predictor itself. It's only available within GastroPlus and the ADMET Predictor module. It's a machine learning model to predict the ELF unbound plasma partition coefficient from the 2D structure. And it was built from data in the literature. The regression model was trained on 56 compounds, with 12 compounds used as a test set to judge the model's predictions on compounds outside of that training set. So the model is based on a small data set, but offers a good initial estimate. The ELF concentrations themselves can be used as a target for optimization and for PD models. And again, I'd like to highlight that this extension applies to the general lung tissue in a PBPK model, and that the mechanistic lung model in the PCAT module already included a lung fluid compartment. Moving on from PBPK to population simulations, I think it's fair to say that there have been a lot of enhancements to the population simulation functionality and virtual BE trials within GastroPlus version 9.8. You can now run automated execution of repeated trials. That is, repeated trials, I believe up to 100 of the same population simulation. So you simply select the number of trials to be repeated and the sample size within those trials. You then define a base name for the output files, and then the population simulator will generate a separate output file for each trial. So you can see we set the number to 10. This was for propranolol, and we got 10 STC files outputted after the simulation. When you're using the load previous option for crossover trials, so you click on the button here and then select those 10 trials that we just generated, you're then presented with the opportunity to include intrasubject variability within your simulations. You can choose not to include intrasubject variability, and then you would get exactly the same parameters as in the previous versions of GastroPlus. But you can choose to include intrasubject variability on input parameters such as transit time, intestinal volume, pH, and the bile salt concentration by selecting the option here. Alternatively, you could apply intrasubject CVs to both the Cmax and the AUC, and we have a box for Cmax and AUC, so you can have different CVs for Cmax and AUCs. Once you've run these trials, you get your standard plot comparing the reference and the test, and if you right click on that plot, you can display the statistics for that trial. But you also get a BEQ file for each trial pair, and this contains the pass-fail outcome, so this trial failed, and it also contains the tabulated results for Cmax, AUC 0 to infinity, and AUC 0 to T. For this example here, we had 10 trials, and it would be quite time-consuming to go through each of those 10 files to look at whether a trial passed or failed. So what we also have is a summary BEQ file for the bioequivalence, and here it lists all 10 trials, so you can see which trials failed and which ones passed, and also the statistics, so the Cmax, the AUC 0 to infinity, and the AUC 0 to T. So there's lots more files that are generated as part of a population simulation. Moving from population simulations to the DDI module. In GastroPlus version 9.8, we've made significant advances in our ability to simulate complex mechanistic drug-drug interactions involving enzymes, transporters, and intrahepatic circulation. In addition, we've committed to provide extensive documentation and GastroPlus model files for our DDI standards database that can be used for your regulatory submissions. Documentation is complete and has been scientifically reviewed for a number of compounds, with many more models very close to being complete. For models that aren't ready to be distributed at the time of the GP8 9.8 release, we are planning to make them available for download as they are finalized, so you won't have to wait for future releases of GastroPlus. We now move on to the many additions and enhancements to our Additional Dosage Routes module. Firstly, yes, we have yet another dosage route that's been added to the additional dosage routes. Here we have the intra-articular route of administration into the knee joint, and you've got the diagram on this slide here. So injections are made into the synovial fluid of the joint. Available dosage forms are solution, suspension, and controlled release, and this is only currently for small molecules. Once drug is dosed into the synovial fluid, the model accounts for the drug dissolution and precipitation in the synovial fluid, diffusion through the synovial membrane, uptake into the systemic circulation from the perfused subintimal layer, and diffusion of the drug into the different cartilage layers. Non-specific binding can be defined as a fraction unbound to simulate the binding to tissue, protein, or hyaluronic acid in all compartments, including synovial fluid, synovial membrane, and cartilage. There are several built-in models to estimate the diffusivity in different compartments. Simply double-click on the empty cell to trigger the model selection, and then you can synchronize across all compartments. Bindings incorporated by this non-specific fraction unbound in each compartment. We have built-in physiologies for healthy human, dog, rat, monkey, and mouse knee articular joint and cartilage, with allometric scaling used to fill any data gaps. Currently, the lymph flow is not utilized, and viscosity is only used to estimate the diffusion coefficient if selected. We have included a new tutorial example for TCA, a synthetic corticosteroid used intra-articularly to treat various joint conditions. And as with all the additional dosage routes, there's a tab in the select curves to probe and fully understand your simulations. And this really unleashes the full power of PBPK. Moving on to the nasal pulmonary compartmental absorption and transit model, which the PCAT module has been in GastroPlus for a fair number of years now, was developed in collaboration with GSK, but just recently we've included two enhancements. One is the lysosomal trapping, and the other one is the addition of a vapor inhalation model. We'll start off with the vapor inhalation model. So this model allows calculation of both absorption from the vapor phase based upon the gas concentration, the inhalation flow, and Henry's law coefficient, or the exhalation for volatile or drug compounds administered through other routes like oral or intravenous. New inputs for the vapor inhalation include the additional flow separate from the deposition model, and this is depicted here on the PCAT window. We have the gas volume and vapor thickness on the physiology tab of each compartment. Back on the PCAT model window, we have the vapor diffusion coefficient, which is a user-defined parameter. We have the Henry's law constant at 37 degrees centigrade, and that's a user-defined parameter as well. And we have the Henry's law correction based on compound properties and the amount of lipids in the lung fluid. Now, this model is really for low molecular weight organic compounds that easily evaporate at room temperature. And for such compounds, the vapor diffusion coefficient and Henry's law constant should be known. And that's these two parameters here, and the program will calculate the third parameter. We have included a new tutorial example for the vapor inhalation, and this is using ethanol. In terms of verification or validation, depending on what term you wish to use, predicting the CP versus time profile following vapor inhalation, we have looked at various compounds, including ethanol, methanol, and acetone, and we need to include and continue that validation with additional compounds, as although the majority are predicted well, some require specific refinements to be made. And here we've got a poster from Jim, who's done most of the work with this new dosage route, and you can have this copy of this poster if you request it from us. The second enhancement to the PCAT model is the addition of the specific mechanism of lysosomal trapping in the epithelial cells of the lung. So this is included by selecting the checkbox, include lysosomal trapping, on the PCAT window. The partition equations are based on our default Lukachova equation. So the plasma and red blood cell parameters are required as a surrogate for acidic phospholipid and protein binding, and they're on the lysosomal KP tab. The lysosome parameters themselves are on the physiology tab. So we have the percentage of lysosomes in the cells, the lysosome pH, the number of lysosomes per cell, and the lysosome membrane thickness. There's limited data for validation of this new enhancement, so we eagerly await your feedback to help us explore the true value of this enhancement. As you can see here, we've got the ability to predict the lysosome concentrations and compare those to the intracellular concentrations. Moving from the PCAT to the OCAT. So now I'm talking about the ocular compartmental absorption and transit model. Here we've included linear protein binding. We've added that to all ocular tissues based on the albumin ratio for each tissue and the plasma protein binding. Previously, we only had melanin binding that was incorporated. Another addition to the OCAT model is the ointment formulations have been added. So drug release from an ointment is modeled by the Higuchi equation, and elimination of the ointment from the surface of the eye due to blinking is also factored in as well. And here we have some of our verification work with fluoromethylone. Moving from OCAT to TCAT. So now I'm talking about the transdermal compartmental absorption and transit model. Dermal dosing is now available for inclusion in MDD files. So here we have on the left an MDD file, and you can see from the drop-down menu, we have the options for dermal dosing, the transdermal. We've also included for the sebum, a new equation for permeability. So this is the Yang and Liang method, and it's based on a wider range of data than the Valivetti method that was originally included. For the intramuscular and subcutaneous dosage routes, we've included a lag time. So this has been added to delay the start of the immune response, such as in the formation of a granuloma. You'd enter your lag time here, and we've also made this plot now active. So if you put a lag time in, you change the parameters A or B describing the equation, then that plot will be updated to reflect the changes in the input. We have added CRU, so that's controlled release undissolved formulations, and this allows you to then simulate for where particles get packed into a core shortly after injection, and over time the particles detach from the surface of the core and dissolve based on the defined particle size and solubility of the API. To account for information, time-dependent changes in the effective depot volume have been included in the muscle and sub-Q tissue. So here we have V0, which is the initial effective depot volume. The inflammation is then described or defined as a tabulated profile or parameters for an equation, and this is the same equation type as for the immune cell layer. Now, at the start of a simulation, you get to choose which setting, either the tabulated profile or the equation, will be used in the simulation using the radio buttons here. Scaling factors can be set for both the muscle and the sub-Q, and these scale the whole inflammation profile either up or down and can be used to distinguish the degree of inflammation between muscle and sub-Q tissue for split injections. In the example here, you can see the muscle, we've set the inflammation factor to 2, and for the sub-Q tissue, we've set the inflammation factor to 0.5. We've also added more additional dosage routes modules to the mixed multiple doses. Transdermal, intramuscular, and sub-Q can now be included in MDD files, and there are some limitations. So intramuscular and sub-Q can be administered together, but not with each other, not with other dosage forms. and the Ocular OC cannot be mixed with other dosage forms currently. Now, before we move on to taking your questions, I would like to acknowledge the team that brought GastroPlus version 9.8 to you. So Simulations Technologies team, led brilliantly by Haiying. So that's Jim, Kay, Jessica, Maxime, and Manas have worked extremely hard, especially over the last few weeks, to get 9.8 ready for you today. They've had a lot of support from our founding scientist, Mike Bolger, and our chief scientist, Viera Lukachova, and also many other teams at Simulations Plus have helped out to get 9.8 to you today. I would also like to encourage you to download version 9.8 as soon as possible. And remember that you can run both versions on your computers. So thank you for your time. And now I guess we'll invite your questions.